Hello and welcome to lecture number 8 of the Cardiovascular section of the Integrative Systems Physiology course. Today we'll be covering capillaries, nutrient exchange, which is of course major site nutrient exchanges at the capillary bed, and the role of the lymphatic system in maintaining cardiovascular homeostasis. So up until this point, we've largely focused on the vascular system, I think, in three contexts. One being out, so what's coming out of the heart in terms of cardiac output, back with regards to what's coming back and with regards to venous return, and then how those mechanical properties of the cardiovascular system actually interplay significantly with cardiac function and can actually dictate cardiac function in a variety of contexts. So the big question at this point is what happens in the interim, and of course that's the nutrient and fluid exchange at the site of the capillary bed. Now, when we're looking at vessel sizes, we go from arterioles to venules with the capillaries in between. You can see here that the capillaries are, of course, the radius is very small, 3 micron radius of the capillaries, very small, roughly a tenth of the size of a first-order arteriole even. So extremely small to maximize diffusion distance, maximize diffusion capability there, minimize diffusion distance. When we start to look in more detail at this microcirculatory circuit, what we can see is that the arterioles have really levels of macro flow regulation, and so there you can see here that you can have changes in resistance at the site of the arteriole, and those are going to alter flow largely through large regions of capillary beds, not necessarily a microflow unit of regulation to get tissue, get oxygen and nutrients to the areas that are most highly metabolically active and relatively heterogeneous tissues. Now, precapillary sphincters, on the other hand, are really this microflow regulation of blood flow. And you can see those are depicted here. So while the arteriole might be altering resistance to a region, and those are going to be the site of regulation of things like sympathetic outflow, for example, these precapillary sphincters are really the microregulatory units that are going to be dictating where blood gets distributed within a given tissue. And so as tissues become more metabolically active and PO2 drops, precapillary sphincters will open up and enable blood flow to go through there to regions that are most highly metabolically active on a micro scale. Now, importantly, when we look at different types of capillaries, there are predominantly 3 types of capillaries. There's the continuous capillary, the fenestrated capillary, and the sinusoidal or discontinuous capillary, and we'll go through these in a little bit more detail here. Now, continuous capillaries are, by and large, the most common type of capillary. They have very small interendothelial junctions that are roughly 10 to 15 nanometers wide, and importantly, they're not very leaky. And so in the vast majority of capillary beds that are throughout the body, certainly within skeletal muscle, within the lung, so on and so forth, there you want to have very tight junctions. You don't want to have a lot of fluid exudation into the interstitial space, but yet you still want to have maximization of the capabilities for diffusion. And so the continuous capillary kind of checks all those boxes as being very thin, but not very leaky. Again, when we look at these from a perspective of a micrograph here, you can see we've got the capillary lumen, endothelial cell, and these tight junctions that are really the holding the capillary together and enabling some fluid and nutrient flux to occur between them, but largely mitigating that leak that would happen under physiologic pressures. Now, in contrast to that, when you look at fenestrated capillaries, here again we have very thin endothelial cells. These are perforated with fenestrations or fenestrae here as depicted in the image to the right. But importantly, these are most often surrounding epithelia. And so when we look at, for example, the small intestine or exocrine glands, such as sweat glands, there you want to be able to have significant increases in fluid flux or transport across these capillaries. And by opening these, having these fenestrae open, that enables the relatively large flux of fluid out of the capillary. Now, finally, when we're looking at discontinuous capillaries, again, these are all realistically across the spectrum of leakiness, if you will. The discontinuous capillary has very large fenestrations and also has very large intercellular gaps. And of course, this is very important with regards to their function in the tissues in which they're found, such as the liver, where what you would like to be able to have is significant flux of fluids and substrates across that, so you can ensure that what you'll be having is substantial fluid flux and the ability to, for example, detoxify and metabolize substrates that are in the bloodstream and get those out of the circulation. So now when we're looking at nutrient exchange at the capillary bed, so one is, what do these capillaries look like? Two, what does the pressure that they're exposed to look like, and how does that interplay dictate how much fluid effluxes out through them? So of course, these capillaries are thin-walled. That thin-walled nature of all of the capillaries facilitates diffusion, but critically, it means that across all of these conditions, you have to have relatively low pressures. And so the predominant pressure drop that occurs in the circulation actually occurs at the arterioles. And so if you recall in that prior figure, the arterioles are the macro regulators of pressure and resistance towards the capillary beds. And that is also under almost all conditions where the largest pressure drop occurs. And that's just shown here in this schematic. So you can see there's a tremendous drop in pressure as you go through the arterioles into the very large cross-sectional area capillary beds, and then subsequently come back through the veins back to the central circulation. Now, importantly, as is noted, even significant arteriolar dilatation or dilation results in relatively small increases in capillary pressure. So you can see here that with arteriolar constriction, for example, in the green line, as compared to normal conditions, we can have a significantly greater drop in this pressure gradient going across that arteriole due to the increases in resistance, and that's going to ultimately reduce pressure further in the capillary bed. But when we have arteriolar dilation, you can see again, we here in the purple, what you can see is that there's a smaller pressure drop here due to the lower resistance. But again, we're not getting very large increases in pressure in the capillary bed. We go from maybe 25 to 35 or so. We're not starting to approach a true arterial pressure because the capillary beds are simply just not made to withstand that type of pressure without having substantial fluid exudation. And so if we look at this in a little bit more detail and quantitatively, if we're looking at, let's say, the effects of arteriolar constriction on capillary pressure, as you can see here under normal conditions, we have a frame of reference at the top. We've got an arteriole, we've got a precapillary resistance, we've got the capillary bed. So again, we're having a substantial drop in pressure across that arteriole, and then postcapillary resistance, and ultimately entering into the venules. You can see very large drop in pressure from the arteriole to the capillary, and a smaller drop from the capillary to the venule. When we look at either arteriolar constriction or potentially venular dilation, both of which would be dictated by metabolic activity within these muscle beds, for example, you can see here we get a large increase in precapillary resistance. That results in a relatively, in the grand scheme of things, relatively small reduction in capillary pressures, but a widening of that pressure gradient here due to that increase in resistance. And again, not a tremendous change in venular pressure, which maintains the driving pressure for venous return through that capillary bed. Now, in contrast, when we evaluate the effects of arteriolar dilation, again, you can see here, normal conditions, arteriolar pressure 60 millimeters mercury, capillary pressure 25, and venular 15. Now we're reducing that resistance at the arteriole and in that precapillary region. you can see that we go from roughly 60 millimeters of mercury in the arteriole, dropping now to 40 millimeters of mercury in the capillary. So significantly closer to what you observe in the arteriole, but again, not approaching mean arterial pressure by any means. And then from the capillary to the venule, now we end up having an augmented pressure gradient here because the capillary is ultimately experiencing this increase in pressure, and we still have a resistance here resulting in a drop in pressure and driving venous return with the pressure gradient from the capillary to the venule back to the right atrium. So overall, capillary beds, again, remain relatively protected from dramatic changes in pressure. And importantly, not only do these tissue beds remain relatively protected from pressure through the regulation of arterial or tone and precapillary sphincters, but of course those precapillary sphincters then end up recruiting more capillaries, and that ultimately ends up resulting in a greater cross-sectional area to perfuse, and therefore dissipates that pressure inside of those tissues and reduces resistance within the capillary. So the way in which this is described is largely through a Crow's tissue cylinder model, and the Crow's tissue cylinder is essentially defined as the volume of a tissue that a single capillary supplies with oxygen or any given nutrient, for example. So you can see that's depicted over here, and so essentially if you have one capillary running through some region of tissue, the region that supplies with oxygen and nutrients is essentially that cylinder and that volume. And you can see as it goes in through the arterial end, highly oxygenated is depicted by red, and then as it travels down the capillary and oxygen exchange progressively occurs, as does nutrient exchange. You can see that you have a relatively deoxygenated sample along with a relatively high concentration of waste products in that capillary at the end of the tissue cylinder. Now, if we take a bunch of these cylinders and add them all up, essentially, you can see that when we have a number of precapillary sphincters closed, we have relatively large definitions of Crow's cylinders here. So you can see that for this capillary that is open, it's applying a cylinder roughly of that size. Similarly, this capillary, which is normally a cylinder away from this one, now has a large cylinder over here and similarly on the bottom. Now, of course, this can occur physiologically with relatively metabolically inactive tissues, and it can also occur pathophysiologically with the loss or blockage of capillaries. So in disease, when you have things like disseminated intravascular coagulopathy, which can occur where you have clots forming throughout the circulation in the early phases of the disease, that can result in the loss of capillaries, recruitable capillaries within some of these regions, and ultimately increase the size of that Crow's cylinder. And what you can think about here is that here, if you're providing oxygen, you're losing an oxygen gradient as you go down the capillary and supply that tissue. You also have a diffusion gradient as you're going radially throughout these tissues as well. So again, this Crow's cylinder model is just depicting the size of some of these cylinders. And of course, when you recruit these capillaries then through largely increases in metabolic activity, you have relaxation of those precapillary sphincters and then opening of those capillaries with subsequent reductions in size in that Crow's cylinder supplying those regions. Now, of course, here not only are you improving oxygenation, at the proximal end here of this, but you're also reducing the size of the cylinder throughout the entire length of the cylinder. So particularly, we're going to have probably the most profound impact is at the distal portions of the capillaries that are providing these tissues and microsegments here. Since you have increases in oxygenation as well as a smaller diffusion distance at the end of the cylinder. So now when we're looking at the factors that actually influence capillary exchange of oxygen and solutes, again, this is really being driven largely by gradients. Okay, and so when we look at, for example, the concentration of free oxygen or free solutes that are in the arteriolar blood that feeds the capillary, that is a major determinant of oxygen exchange within any given bed. So for oxygen, that is the partial pressure of oxygen, which we'll talk about calculating later. But of course, you've got the partial pressure of oxygen, you've got the oxygen content of the blood. So this is free oxygen. This is how much oxygen you actually have in the plasma plus hemoglobin, right? So as you offload some of this out of the plasma, then you will be backfilling that with oxygen from the hemoglobin. Capillary flow, of course, is a major determinant, as we discussed in our last discussion section. With increases in cardiac output, again, you can increase capillary flow and increase overall delivery to those regions of metabolic activity, and the radial diffusion coefficient. And so here, we'll assume that this radial diffusion coefficient is the same for the blood, the capillary wall, the tissue, and all of the entire length of the capillary. And so really just one large homogeneous radial diffusion coefficient for all of these samples. Finally, we've got the capillary radius. So if you have a larger capillary, that's going to be able to bring more substrate in to diffuse through. The radius of that tissue cylinder that the capillary is supplying, right? So how far does it have to diffuse, and what type of gradient are we looking at enabling to promote diffusion? And then the oxygen consumption by the surrounding tissues or nutrient consumption by the surrounding tissues. An axial distance along the capillary. So how long is that capillary? So when we start to look at the diffusion of, for example, small water-soluble solutes across the capillary wall, we're going to define that as the flux of a solute that crosses the capillary wall, or flux is defined as J sub x. And so here we've got the flux of any given water-soluble solute is defined by the permeability coefficient times that capillary to interstitial fluid concentration of that water-soluble solute. Okay, so again, not really pulling any punches there. It's about as straightforward as it seems. The flux is defined in moles per centimeter squared per second. And so it's realistically very difficult to know what the surface area is for either a specific capillary or even for a specific set of capillaries or a capillary bed. But if we just take this and depict it in schematic form here, you can see that we've got flux across a given capillary wall here. We've got the surface area, the diffusion coefficient, defining this as a very homogeneous microenvironment here, both the blood, capillary wall, and tissue having the same diffusion coefficient in most instances would be the core assumption there. The concentration of the solute in the capillary and the concentration of the solute in the interstitial space. So you'd be looking at flux per unit of area, and of course a major determinant of that would be the diffusion coefficient of the capillary wall. So now on the one hand you've got the diffusion coefficient across that unit of area. When you start to look at the mass flow of the solute transferred per unit of time, then we're viewing that as being flow as Q sub x. And so what we'll be doing there is taking that whole surface area times the flux to ultimately define the mass flow across that unit of area. For the extraction of a solute, we've got the extraction ratio, so that is very similar to the arteriovenous oxygen difference, except it just expressed as a ratio now. And so you've got the arteriolar concentration minus the venular concentration divided by the arteriolar concentration. So essentially identical to the arteriovenous oxygen difference, but only expressed as a fraction now. Of course, there's a dependence of this extraction ratio on permeability, surface area, and flow, and the extraction ratio will be proportional to permeability, proportional to surface area, and inversely proportional to flow. But the only thing I'd like you to take away from this table here is just that permeability coefficients do vary for different solutes. Again, I think if you're incredibly fascinated by transport of solutes, it's important for you to probably appreciate this in more detail. But for the purposes of the course, really all I want you to appreciate is that there are differences in permeability coefficients for various different solutes. And of course, as you generally have the diffusion of the solutes, you will also have a capillary exchange of water along with them. And so the capillary exchange of water is given by the Starling equation. And that is depicted here. So if we take this and break it down a little bit further, so we've got, again, flux being J sub V, volume flux of the water across the capillary wall. So this is centimeters cubed of water per centimeter squared of surface area per second. And what we have as defining characteristics of this equation would be L sub P, which is the hydraulic conductivity, or a measure of capillary leakiness for that given bed or that given region of interest. The capillary hydrostatic pressure, or the P sub C, so capillary pressure, that's in millimeters of mercury, really depicting intravascular capillary pressure, or upstream driving pressure from the hydrostatic pressure difference. We've got an interstitial fluid hydrostatic pressure, or P sub IF. And so again, that is the downstream interstitial fluid pressure. So that difference between inside the vessel and outside the vessel being that hydrostatic pressure difference. The capillary colloid and osmotic pressure within the vessel, of course, that is, and within the capillary, that's caused by plasma proteins. Outside of the vessel, you have the interstitial colloid and osmotic pressure. So again, free proteins within each region of this capillary point of exchange. And then finally, a colloid as osmotic pressure reflection coefficient, which describes how well a semi-permeable barrier such as a capillary excludes or reflects solutes as that water moves across that gradient. So you've got a pressure gradient, you've got a protein concentration gradient, and a reflection coefficient here, ultimately, which is going to dictate your net filtration pressure across a given capillary or capillary bed with a conductivity coefficient there. So when we look at that's all fine from an equation perspective, but then in reality, what really happens across that capillary bed? And so when we're looking at typical transcapillary driving forces for fluid movement, you can see that as we are at the proximal end of the capillary, nearest the arteriole, we have net filtration out of the capillary into the interstitial space. And as we get towards the distal end of the capillary and nearer to the venule, we have net absorption into the capillary. And critically, it doesn't take too astute of an eye to notice that the two triangles here are quite drastically different shapes. And critically, at any given capillary, there's a net filtration out of the capillary, which is a net loss of fluid into the interstitial space, despite the fact that we have some absorption occurring here at the distal end of the capillary. And so the big question is, well, where the heck does all that go? And the reality is that we do have to have a system to deal with that fluid exudation into the interstitium, and that fluid is returned to the circulation via the lymphatic system. And so that is just coarsely depicted here. So with the lymphatic system, you end up having about 2 to 4 liters per day of flux coming through the lymphatic system, and importantly, you can see as depicted here, with 2 to 4 liters per day of fluid coming through the lymphatic system. That's roughly at the higher end. That's starting to approximate what your total cardiac output would be, or what your total cardiac volume would be, right? So you have about 4.5 to 5 liters of blood in the average 70 kilogram male. And so when we look over here and we say, okay, so per day, and with regards to cardiac output, the average person has about 7,200 liters per day of blood pumped out of the heart. That comes here through the arterial system into the capillary beds. What we have is actually a net exit from the capillary of about 20 liters per day. Again, net entry into the capillary in the distal portion of 16 to 18 liters per day, with that difference being 2 to 4 liters per minute. That ends up having 2 to 4 liters per day coming back into the lymphatic system and being returned to the circulation via the lymphatics. Otherwise, this would simply result in progressive exudation of fluid into the interstitial space and massive edema. Now, when we're looking at lymphatic system filling and emptying, of course, there are two phases here. One being the expansion phase of the lymphatic filling, and so that is when the interstitial pressure is exceeding the lymphatic pressure inside of the lymph node. There, the lymphatic valves are open, and that enables filling of the lymphatics. And then during the compression phase, the interstitial pressure is less than the lymphatic pressure, and that ends up driving the lymphatics valves closed, and the fluid is then pushed from the initial lymphatics to the collecting lymphatics, and that can occur with a variety of sort of even just normal events that are occurring within the body, which would include intestinal peristalsis, respiration, skeletal muscle contraction, even subtle muscle movements are going to result in the compression and pushing of lymph fluid and water back into these collecting lymphatics, and those collecting lymphatics are going to drain into the venous system, ultimately returning that fluid exudate from the capillary to the interstitium back into the circulation and preventing you from having substantial increases in interstitial fluids. So, of course, that broaches the question, well, what happens when these things are a little bit abnormal, right? And what you end up having at that point is edema. When edema becomes severe, you can have an entity called pitting edema. And so what you can see here is that this is a commonly used clinical test for severity of edema. If you push down on the front of a person's calf, which generally should be relatively taut and not filled with interstitial fluid, when you push down all the way to the bone and then remove your finger, you can have this process called, this observation called pitting edema, where a pit is left behind. That can be left behind for a short period of time, or that can be there for a couple of minutes, depending on severity of edema. And then for most patients that have severe edema, then you would either be assessing the patient for obstructions to outflow, or for assessing the patient to try to determine their volume status. For example, with patients with heart failure, if they are starting to retain too much fluid, have substantial peripheral edema, then you'd administer a diuretic to try to optimize their fluid status. So overall, in summary, again, it's pretty straightforward to start off. The left ventricle pumps blood into the vasculature. That blood goes from the arteries to the arterioles to the capillaries. We've got gas and fluid and nutrient exchange occurring in the capillaries. Any of that net fluid exudation that occurs in the capillaries is returned to the circulation through the lymphatic system, and of course the blood is then returned from the heart, from the capillaries to the venules, to the veins, back to the right atrium. So hopefully this is all coming together at this point, and I'll look forward to seeing you in the next lecture. Thank you.